Awesome. Well, hi, Sister Mayo. This is Catherine and Holly, our new group, and we are working on this lesson 7 group work problems. And Holly would take this off with number 1.
Okay, so in the first one, they give you just different viral genomes that were examined in the laboratory, and they wanted to know what each one contained, the different structures. So they wanted to know if it was double-stranded or single, RNA versus DNA, or circular versus linear. So for virus number 1, for the first part, I got that it was RNA, double-stranded, and circular. So the difference I could tell between the RNA and the DNA was having a U or a T. It shows the percentage. Then for the double-stranded, it from being resistant to the exonuclease but sensitive to the endonuclease, then that's why it would be double-stranded. And then the circular, because it, or the, and then with the percentage, then you would know if it was circular, or circular and linear is because of the sensitivity and what it's resistant to, and then the double-stranded came from how far apart each percentage is. So then I just went through that for each one. So then number 2, RNA single-stranded linear. Number 3, DNA double-stranded linear. And number 4 was DNA single-stranded and circular. Those are my answers.
So for problem number 2, it's asking us to fill in the blanks. So for number 2, part A is, I can never say this word, topoisomerase 2 is composed of 4 protein subunits and uses ATP to compact bacterial chromosomes, while a nucleosome contains 146 to 147 base pairs of DNA and 8 histone proteins. For B, the DNA double helix is 2 nanometers wide, and adjacent base pairs are separated by 0.34 nanometers in the DNA double helix. For part C, topoisomerase 1 introduces positive supercoiling into DNA and cuts 1 DNA strand. D is the origin of replication in E. coli is called ORIC. For E, in the Hershey-Chase experiment, bacteriophage T2 proteins were labeled with a radioactive isotope of 35S, while bacteriophage T2 DNA was labeled with a radioactive isotope of 32P. And then for F, the sugar in uridine is ribose, while the sugar in deoxythymidine is deoxyribose. And we just found these answers in the instructor notes.
Yes. So then for part 3, they say, suppose a translocation occurs on chromosome 14 and chromosome 21 in an individual. The translocation is the result of the fusion between 2 non-homologous chromosomes. Chromosome 14 is 109 million base pairs in length, while chromosome 21 is 48 million base pairs in length. For this problem, assume that the translocation fused the entirety of chromosome 14 and the entirety of chromosome 21. So the part A, they want to know how long DNA portion is of the translocated chromosome in meters. So I started out by adding them together because it says that it's their entirety. So I did the 109 million base pairs plus the 48, and then I got 157 million base pairs. So then they want to know what it is in meters. So they give you a conversion between the base pairs, and then, as Catherine mentioned, the 0.34 nanometers, and then you use a conversion factor, and there's really just a couple or multiple different things to get to meters, but I just used the nanometer, 2 meters, and I got 0.0534 meters. Then in part B, they want to know how many nucleosomes are found within the translocation of the chromosome. So I took the answer that we got when we added them together, so the 157 million base parts, and then our base pairs, and then they gave us the conversion factor of 195 base pairs to 1 nucleosome. So then I multiplied those, and I got 801,020 nucleosomes. So then part C, they want to know how many of the H2A molecules would you expect to find in this translocation on the chromosome. So we know that for every 1 nucleosome, we have 2 H2As. So I just multiplied my answer by 2 from part B, and I got 1602040 of the H2A molecules. So then for part D, they wanted to know the range of the radial loop, and they gave you 2 different ranges where the first one was 2,500 base pairs, and the other one was 2,200,000 base pairs. And so then I just took the total of the base pairs that we got, divided it by the upper, divided by the lower, and then I got that the radial loop range would be from 785 to 6,280.
So for problem number 4 is kind of similar to 3, but it says the bacterium Mycobacterium tuberculosis has a single circular chromosome that is 4,410,000 base pairs in length. If the chromosome was stretched out linearly, how long would it be in micrometers? So pretty much what you do with that is I started off converting it to, can you see that? I don't know if you can see that.
Come out just a little bit.
There it is.
Yeah, there you go. That's better.
So for number 4, for part A, what I did is I converted the 4,410,000. Well, I converted it to scientific notation just to make it easier. And then you multiply that number, 4.41 times 10 to the 6 times 0.34 nano, because that's the conversion that we were given, over 1 base pair, as you can see here. And then you convert that into micrometers, and there's 1,000 micrometers for every 10⁶ nano. And then after you multiply all across, you get, and divided by the 10⁶ nano, you get 1,499.4 micrometers. So for part B, it's asking, what is the diameter of the M tuberculosis chromosome in micrometers? And then the circumference equals pi diameter. And then for that one, all you have to do is take 1,499.4 and divide that by pi. And then you get 477.2 micrometers as the circumference. For part C, it's asking, suppose the M tuberculosis genome is organized into loop domains, with each loop domain contains 50,000 base pairs. If one of these loops was stretched out linearly, how would it be in micrometers? What you do for that one is you take the 50,000 base pairs and you times it. You do the conversion, you times it by 0.34 nano, and then you times that times 1,000 micrometers, and then you divide all of that by 10⁶ nanos, and is equal to 17 micrometers long. And then for part D, it's asking how many histones are found attached to the M tuberculosis chromosome. And we got 0 because histones aren't present in bacteria. They're only present in eukaryotes. So here's, never do it right. Okay, there's my work.
Okay, so then for 5, they gave you the different pictures of the different substances or molecules. Let's see exactly what they are. They give you the different structures of nucleosides and nucleotides, and they want you to identify which ones are nucleosides in the first one. So your nucleosides are going to be the ones that don't have the phosphate group. So that would be A, E, and F. Then the nucleotides will have it. So that will be the other 3, which are B, C, and D. The ribose, you would know from having the RNA, which would have a U instead of a T, and that would be C, E, and F. Then the deoxyribose would be A, B, and D. And then they want to know the pyrimidines, which would be C, D, E, and F.
Awesome. And that was our group meeting, and cool.